Good morning, students. Now we're going to the second part of the protein synthesis, that is the translation. For translation, remember, the mRNA that is formed or the mRNA after the formation that has come out from the nucleus or was in the cytoplasm. Okay? From mRNA to the formation of protein is translation. Clear? Now, what happens to the mRNA that comes out from the nucleus? In that case, what happens? This is the ribosome. Okay. So, this is ribosome. Ribosome has two subunits, and in between these two subunits, the mRNA remains. This is mRNA. Clear? Now, we'll be going step by step what happens in case of translation, which is one of the most longest and important part of the protein synthesis. First is the activation of amino acid. Now student, you see one thing, when I speak about the activation of amino acid, I am writing here, activation of amino acid, number one, activation of amino acid. What happens here? Amino acid, I am writing here, amino acid need to be activated. That means it need to react with ATP. When it reacts with ATP, then we call it activated amino acid. Then it is charged. It is called what? Activated amino acid. And that activated amino acid can now bond with that of the and come and link with that of the tRNA. So to bond with the tRNA, the amino acid that is present simply it can't come and join with the tRNA. That need to be charged. That need to be activated. How it is done? When it reacts with ATP. That is the first one. Clear? Now we will be going for the initiation of the protein synthesis. Now see one thing. This is a two subunit of ribosome. This is 30S and this one is 50S. Ribosome, and in between there is the occurrence of the mRNA. And these are the nucleotide. We are finding it has 3 nucleotides, 3 nitrogenous base forms the triplet, is not it? So, this is mRNA. The ribosome has 2 sites. This one of these is called A site. A site, SIT site, and other one is called the P site. A site means amino acid binding site. What is called? A site is called amino acid binding site. Amino acid get bonded there. Another is the second one is the P site. This one is what? P site. What is P site? Peptidal site, because the peptidal chains are formed there. This is called what? Peptidal site. Now, I am rubbing this one now for going to the next part. See here, tRNA will be coming and getting linked here. This is tRNA. Another thing comes here. See, tRNA will be bonded with that of the mRNA that is present based on the codon and anticodon. What is that codon and anticodon we have done earlier? See, 3 triplets. 3 nitrogenous base together. 3 base. Nitrogenous base is called triplet. Okay. They are called what? Codon. 3 nitrogenous base, they are called what? Codon. So I am taking these 3. These 3, suppose here these 3, these are what? Codon. And the tRNA that will be sitting over that of the mRNA. Try to get my point. tRNA that has come and will be sitting over that of the RNA need to be complementary with that of the codon pattern. So that complementary here, in that case it is called the anticodon. What is that meaning? Suppose here A, G is there in case of the mRNA. The incoming tRNA need to be T, T, C. Can you get it? So this is codon. Why it is present? It is in mRNA. This is anticodon. Why it will be? This will be tRNA. Understood? Now tRNA is there. So the amino acid will come and bond here. This is one thing. Now this is the first amino acid that has come. First amino acid will be based upon the specificity. What is the specificity? Specificity will be based upon the tRNA having the unpaired nucleotides. Okay, tRNA has unpaired nucleotide at the top. So, based on that, see, suppose this is the tRNA. Here, there is the occurrence of unpaired nucleotide. And the coming amino acid will be bonded based on the unpaired nucleotide that is present there. Understood the thing? So, this first amino acid has come. But the second one will be based on, I am rubbing this one. Second one that will be coming, this is the first one. Second amino acid that will be coming, third, fourth that will be coming, will be based on the acid base interaction. What is the meaning of that? COOH and NH2 bonding. That means amino acid has two parts. One is the base part, other is the acid part. So if the acid part of the amino acid is spotted outside, incoming amino acid will have the base part undergoing reaction with that. So that way, first has come, second, third, fourth, fifth amino acid will be bonded and there will be occurrence of chain of amino acid. Clear? Now coming to the initiation of protein, how that process starts. Process of amino acid chain leading to the formation of protein or the occurrence of the starting of protein occurs when there is the coming of the triplet AUG. How it comes? This is mRNA. mRNA is gradual in the process of sewing machine and the stitching machine that we have in the home. The cloth keep on moving, is not it? Like this. mRNA also keep on moving. Moment there is the coming of three triplets, I am here. Suppose it has come here AUG. What is it has come? AUG. Moment the AUG has come, then the protein synthesis starts. So, what is the initiation codon? Initiation codon is AUG and it starts the process. Clear? So, that is the initiation of protein synthesis. Now, coming to the elongation of peptidal chain. As I told you, one amino acid get linked with the another amino acid based on the acid and base interaction. That is the one thing. Second thing, try to get my point. Moment the chain of amino acid is formed, it may be by 4, 5, 6 amino acid. Then what happens? The amino acid from the A site is shifted to the P site. From here it goes to P site. Got it? This is A site. This is P site. So chain of amino acid will be formed where? In the S site. Moment 4 to 5 amino acid chains or the amino acids are formed to form a chain. It is shifted from A site to P site by an enzyme. It is called transferase. Transferase 1. Understood? Where it shifts, it takes it to the P site. Got it? What happens in the P site now? The complete maturity and the occurrence of the peptidal chain, peptidal bond formation occurs in the P site. Clear? Now see another thing. I am rubbing this one. This is the amino, this is the P side. Now, from A side, the chain has come here. Isn't it? A side is open. New amino acid will be coming here. Who has changed this? Enzyme. What is the name of the enzyme? Transferase 1. Moment the peptidal bond and the peptidal chain formation has been completed, again it will be shifted to A site. Understood the thing? Now what will happen to the chain of amino acid that has come newly? There is a simultaneous process. Moment P site shifts the peptidal chain from P site to A site will be shifting the chain of amino acid from A site to P site by transferase 1. That is by shifting of the peptidal chain from P site to A site by transferase 2. So, two enzymes are working here. One is transferase 1, another is transferase 2. What transferase 1 does? Transferase 1 shifts the chain of amino acid, forming a chain from A site to P site. What transferase 2 does? It shifts the peptidal chain from P site to A site, and that is a simultaneous process. Both the processes keep on going at the same time. Now coming to the last one. Termination of protein synthesis. Like that, the initiation codon, AUG, there are also terminating codon. We have found earlier. What is the name of the terminating codon? UAA, UAG, then another is UGA. Out of these three, any one, if it comes now below UAA, suppose, if it comes below the tRNA, that is in the A site, got it? Then protein synthesis comes to stop. When that is switch on, then it to be switch off. Switch on, one given by AUG. Switch off will be by UAA or UAG or UGA. It comes to stop. Peptidal chain formation come to halt. But till now there is no occurrence of protein. Finally, what happens? The protein is formed. That means from here the peptidal chain that is formed need to come out. Coming out of the peptidal chain is the occurrence of protein, and it is helped by a factor. I am writing down the factor F1, factor F1, F2, and releasing factor R1 and R2. It helps in taking out the peptidal chain from the A site, and when it comes out of the ribosome, then it's a protein, just like a potter making clay materials. Up till when it is in the wheel, you cannot call it to be a tub or any sort of mark or defined material. Moment it is taken out, then you call the utensil by giving a name. Same thing happens in case of the protein synthesis. Till it is here, you are not calling it to be a protein. Protein is not formed. Releasing factor R1, R2 and F1, F2 then takes out the peptidal chain from ribosome. Moment it comes out, it is protein. That's how the protein is formed. So, transcription and translation makes the process of protein synthesis a complete one, and formation of protein occurs. Thank you, students.